Hello, welcome back for part 5 of our look at cell biology. And we're going to focus on this video on the second half of um, protein synthesis. All right, and I think we left off with the idea of, um, you know, with transcription, we're taking a segment of uh, our genetic material of a chromosome, and we are replicating only that small portion. And that small portion represents what we call a gene. And it is these genes that code for specific proteins. And so we need to kind of pick up from here and continue on our way of trying to understand now that we have messenger RNA, right, now that we have taken the DNA strand, now that we have taken this DNA strand and have translated it, or transcribed it, sorry, and transcribed it into a pre messenger RNA format. And that pre messenger RNA format, remember, was made up of a series of introns and exons. So you might have an exon and an intron and an intron and an exon and an exon and an intron. And if you remember, we remove the introns, and what we are left with then are the exons. And it's these exons that code for the amino acids. Now in this case, these exons would be, in this case here, no, I lied, U, A, C, C, A, U, G, C, G, U, T, I'm sorry, U, A, and I'm just going to put a G here to make that a C. And so these exons now code for the messenger RNA, and it's this messenger RNA, remember, that is going to leave the nucleus and heads to the cytosol. All right, and so let's go ahead and let's pick up with that next. So translation takes place, it happens at the site of the ribosome, of the rRNA. Right? And so you can see here that our rRNA actually contains two parts. Right? Our RNA has two parts. And so I kind of drew that on the last video. It's got this smaller subunit, and then it's got a larger subunit. The smaller subunit has a measurement of, if you could measure this, of 40S, and the larger subunit has a unit of measurement of 60S, and the whole ribosome has a measurement of 80S. And that S that you see there, that S stands for Svedberg. It's a Svedberg unit. Svedberg was I believe he was, I don't even remember what nationality he is now off the top of my head. But he was a researcher that looked at density, and what he proposed was The density of an object is going to determine how fast it falls through a column of water. And so, uh, because the smaller subunit, because the smaller subunit um, is lighter, it took longer to fall, and so that unit of measurement is a smaller unit. The larger subunit is a 60s because it fell quicker. So the faster it falls, the higher the Svedberg unit. And so that's why when you combine the 60 and the 40s subunits, you get 80s. All right, so this is not a cumulative thing, but this is based on density of the whole thing. So the whole ribosome is an 80s. And again, that's where we get that. That's what that S is standing for. It's a Svedberg unit. All right. But the other things that we need to consider here at the site of translation is not only the rRNA, but we've also got codons. And so we take the messenger RNA and we actually divide that up into 3 nitrogen base pairs. Right? And each collection of 3 nitrogen base pairs is a codon. And that codon codes for an amino acid. Right? So the codon codes for the amino acid. Now, how does the cell know which amino acid to bring to the site? Well, it reads the code, the rRNA reads the codon of the messenger RNA, and it attracts a tRNA, the transfer RNA, and it contains an anticodon. What's an anticodon? It's the codon opposite of a codon. The anticodon is the code that is opposite of the codon. So let's say you had let's say you had A, U, G, C, U, A, C, C, G, U, A, C. All right, this is your strand of messenger RNA. Well, 3 bases, that's a codon. 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 So each of these is a codon. Each of these is a codon. And it's a codon that is going to be read by the rRNA. Well, the anticodon of AUG, the anticodon of AUG would be UAC, because opposite of A is U, opposite of U is A, and opposite of G is C. And so this is the anticodon. And each of these codons is going to have an anticodon that is opposite of it, that is associated with a tRNA. Right? So let's kind of look at how this all plays out. All right? So I'm gonna again, I've no consistency with my string of RNA, messenger RNA. So please don't get frustrated with that. So I'm going to draw in my small subunit here. I'm going to put in my large subunit right there. Now, I'll kind of mention this now because I haven't mentioned it yet, but notice that there's these spaces, right here, right here, and right here. Those spaces actually have names. They are there intentionally. Right? This first space is what we call the A site. Right? A for acetyl. Right? That's the acetyl site. The second site is the P site. Right? It's the P site. P for peptidase, right? Or peptide. And then this third site is the E site, and E is for exit. All right, E is for exit. So you've got an A site, a P site, and an E site. All right. So let me go ahead and let me put in the messenger RNA. All right. So what ends up happening is our strand of messenger RNA will come in, and the first codon attaches to the small subunit, and then the P site, or I should say, then the large subunit comes into place. Right, so our first codon is in place. Now remember, there's a bunch of other codons that are behind it. So this is the whole string of messenger RNA. Okay, so here's our messenger RNA. It is going to, again, the first codon is going to come in. It's going to bind to the A site. And the A site is going to say, okay, AUG. What tRNA do we need? Well, here's a tRNA. I'm going to put in a bunch of tRNAs here. Here's a tRNA. Here's a tRNA. So I'm drawing some of these tRNAs. At the tip of each of those tRNAs is an amino acid. I'm going to make all my amino acids blue just for the sake of simplicity. And at the bottom of each of these tRNAs, there is an anticodon. Maybe a UAC, maybe a GGG, maybe a ACG, maybe a CUU. All right, so each tRNA is going to have an anticodon that is associated with it at one end and the amino acid at the other end. So the anticodon of AUG is going to be UAC. All right. So here's UAC. All right. This tRNA with this amino acid is going to come into the A site. So let me redraw that. This is where it's going to get really ugly. This works a lot better in the classroom. Just saying. Okay. Now, everything here is now going to slide one position. Everything is going to slide one position. So the UA or the AUG is now going to come here. The CCC is now going to come here. Right? Everything shifts to the left. Everything shifts to the left. Right? So if I redraw, I'm going to redraw this really quick. And so here is now the AUG. CCC is there, and then following that will be the rest of the messenger RNA. And I'm going to put in Oops, that's not the color I want. All right. So there's that tRNA with its amino acid. And then we need the anticodon of CCC, which is going to be GGG. And so this amino acid is now going to come over into the A site with its amino acid. And this is where we start to now see peptide bonds being formed, because this amino acid and this amino acid become attached. In fact, this amino acid is going to be joined to this amino acid. And there's an enzyme that directs that process. The enzyme that is going to create the peptide bond between the amino acid in the P site and the amino acid in the A site is called peptidyl transferase. Let me try rewriting that. Transferase. There we go. All right. Peptidyl transferase is the enzyme that is responsible for attaching this amino acid to this amino acid. Now, once that has done that, everything shifts to the left again. So everything moves down. Now, this tRNA no longer has that amino acid attached to it because it's attached over here. So this tRNA leaves. Right? It finds another amino acid. The CCC is now in the second space. The UGC is now in the first, is now in the A site. And the process starts over again. Another tRNA comes in. When that tRNA comes in, peptidyl transferase is going to come in. It's going to take these two amino acids and now attach it to the next. And then everything shifts. And then it's going to attach the next. And then it's going to attach the next until you get this linear sequence of amino acids. I'm going to post some videos in the module as well to help you visualize this more. This does not do well in a static environment. In other words, there's lots of movement to it. But how do we know? How do we know what amino acid is actually there? Well, good question. All right. There is a amino acid table. All right. And so this table tells you what codons code for what amino acids. Now, it's clear that you understand this. This is important. It is the codon that determines or codes for the amino acid, not the anticodon, but it is the codon. In fact, we call this the codon table. Right? We call this the codon table. So if you look, AUG, right here, AUG codes for the amino acid methionine, which, by the way, is one of the essential 9. All right. If you look over here, CCU codes for proline. All right. UAU codes for tyrosine. There are 3 codons that code for a stop codon. This tells the rRNA protein is done. Also notice that there is redundancy that's built in. In other words, if you look here, like there are 6 codon sequences that will code for leucine. There are 2 codon sequences that will code for histidine. There are 4 codon sequences that will code for threonine. All right? There's redundancy built into the system. 4 nitrogen bases, but we're grouping them in groups of 3. that creates 64 possible amino acids for which we only need 20 or 21. So that redundancy helps to stabilize the protein structure. So let's say there was a mutation that happened going from transcribing from DNA to RNA. Let's say for some reason instead of a um, instead of an ACU, let's say there was an ACC that was present. Well, that point mutation still codes for the same amino acid. Every single amino acid has a duplicate, at least one redundancy, except AUG. There's only one start codon, and every single messenger RNA strand must start with AUG. Every single messenger RNA strand must start with AUG. That's how the rRNA knows where to start building the protein. So that's important for you to recognize. Now, to help you figure this out, I do give you this slide, and this slide has some practice on there. All right, so up top here, the example is worked out for you. All right, and you can now come down here and you can practice filling that in and see if you can actually come up with the right amino acid code. All right. This is something that we can look at during class or the Q&A sessions. On the exam, you will be given the codon table. You are expected to know how to use it. All right. So that is something that will be on the exam. Now, what follows is a series of images that are going to help you try to visualize this process. So take your notes that you've been able to copy down from the past 15 minutes or so and see if you can apply it to the process. Right? See if you can apply it to the process. So over here, this is transcription. This is going from DNA to the messenger RNA. Right? Here is the translation. This is now in the cytosol. This is the rough endoplasmic reticulum, or the rRNA. And this is the messenger RNA that's now being translated. Right? You can see the A site, the P site, and the E site there. And again, there is a process here. Right? So you can see everything has shifted over. tRNA comes in. This amino acid gets added to this chain. Everything shifts. This tRNA leaves. This tRNA is now in the P site, and a new tRNA will now come into the A site. All right. Once you hit a stop codon, there's what we call a releasing factor that will come in and actually release the messenger RNA. It'll release the messenger RNA from the rRNA. Right? And once that happens, you've got your protein that is formed. Right? This is a primary protein structure. This is a linear structure of amino acids that are linked and joined by peptide bonds. And the messenger RNA gets broken down. The messenger RNA gets broken down. Right? This is just a visual of what tRNA looks like, in case you were trying to wonder what I was drawing on the previous slide. You can see the anticodon is at one end, and the corresponding amino acid is at the opposite end. All right? And again, this is another visual of that whole process, right, with transcription happening inside of the nucleus and translation happening inside the cytosol. So you've got a couple different visual perspectives for you to consider. And then on this slide here, you've got, again, step by step what is happening. All right, you have step by step what is happening. The last slide that I want to kind of talk about on this has to do with what happens after we have that primary protein that is formed. We know that the job of the rough endoplasmic reticulum is to transport the protein. All right, well, where is it transporting it to? Well, what it does is it packages it inside of a little vesicle, all right, and um, it sends it to the Golgi body or the Golgi apparatus. And it's the job of the Golgi apparatus or the Golgi body to then go ahead and actually pack the protein. So it takes the protein from a primary protein to a secondary protein to a tertiary protein where it may be active, and then we'll go ahead and eventually send it to, um, if it needs to, creates a quaternary protein, right, if needed. But remember, if it's a linear protein, it can be active as a tertiary protein. If it's an enzyme, it can be active as a tertiary protein. But it's the job of the Golgi body to go ahead and do that. And the Golgi body actually has two sides to it. So if you look at the structure of a Golgi body, if you remember from the lab, all right, so the Golgi body is this stacked, flattened, it's a flattened stacked of compartments. Right? This is what is known as the Golgi body. All right, and when that happens, when that happens, the protein coming in on its vesicle, so this is the transport vesicle, that transport vesicle binds to the Golgi body. This is called the cis end of the Golgi body. That linear protein, all right, will then go ahead and be released into the Golgi body. The Golgi body will go ahead and packages it, ending up as either a tertiary or quaternary protein. And then on the opposite end, it gets put into another secretory vesicle, and it gets sent to wherever it needs to go, whether it's in the cell or the body or some other organ or tissue. Well, this end is called the trans end. Right? The cis end and the trans end. And once that happens, the protein is active, it's functioning, and it is released. Um, and so that is the end of protein synthesis. That is the end of protein synthesis. We've got 3 more videos that we're going to go through and do. And so stay tuned, and I'll catch you on the flip side.